sexes, and then later some kind of modifier evolves to suppress that trait. So this might be really important in intralocus sexual conflict, if conflict in males and females over this monomorphic gene expression for horn genes then causes sex limitation. By contrast, maybe you might avoid sexual conflict if you evolve a sex-limited trait from the outset. So if there was an origin of a male or female specific allele, you could get a sex-limited trait immediately without ever passing through this intermediate monomorphic state. So I'm really interested in asking, how frequently do each of these models occur? Do you need to go through the 2 steps that the silencer model would suggest, or can you really evolve a sex-limited trait immediately? And second, how do these alleles become sex-specific? So what's making a monomorphic versus a dimorphic allele? So I think we can really answer these questions in the Drosophila montium subgroup. So this is a subgroup of fruit flies with over 76 species. I'm just showing you a subset here, but I think what's really exciting about them is that females come in 2 different color forms in some of the species, but not all. By contrast, males are always a single color. So some species have males that are monomorphic for dark pigmentation, others have males that are monomorphic for light pigmentation. But whenever we've done crosses, us and others, we've shown that this is usually controlled by an autosomal locus with 2 alleles, and usually the dark allele is dominant in everyone but Drosophila javelina. So I wanted to ask, what is this Mendelian locus controlling female pigmentation? And I started by studying Drosophila serrata. So we used an introgression mapping approach where we took females from a line that only produced dark females and crossed into males from a line that only produced dark males, and hoped that with many successive generations of backcrossing, we'll be left with what's hopefully the locus that's been refined through recombination. So what we did was we genotyped nearly 80 flies from light females and dark females from this introgression line for 40 markers. I'm showing 15 here. What you can see is wherever there's so the columns are markers and the rows are females, and so every place there's a yellow box, it means there's a homozygous light parental allele, and everywhere there's a dark box, it's a heterozygous. So you can see that right within this blue box, there are markers that are perfectly fixed and associated with female color. However, 708 kV is still a large window, and so we wanted to refine this a little bit further. So we teamed up with Adam Reddix and Steve Chenoweth at the University of Queensland, who have developed a panel of 100 inbred Drosophila serrata lines with whole genome sequences. So we were able to use this panel to look for an association with female pigmentation. And very fortunate for us, our 2 best candidate SNPs occurred within the same interval that we had mapped with our introgression mapping. And our top SNPs, after Bonferroni correction, both occurred in the intron of PDM3. So what is PDM3? PDM3 is a PIT-OCT-UNC gene transcription factor that has 2 binding domains, shown here in yellow and purple, that together bind to regulate DNA. It was discovered in 2008 and has since been shown to be involved in olfactory development and brain development. But here I present some data showing that PDM3 also represses pigmentation in Drosophila melanogaster. So I manipulated gene expression by either knocking it down or overexpressing it, and found effects on pigmentation. So in panels C and D, you can see that when I use RNAi to knock down PDM3 levels, flies get darker compared to the controls, females and males on the left. Similarly, if I look at a line where there's been a transposable element inserted into the yellow binding domain that I showed you previously, you see an increase in pigmentation as well, compared to flies where we've precisely excised the transposable element. So this suggests that PDM3 in melanogaster is acting to decrease pigmentation and normally represses it. But I can do the opposite experiment and try to add more PDM3 when normally there's not. So if I increase PDM3 and drive expression to the posterior segments here, what you can see is they're much lighter. So again, this suggests that PDM3 in melanogaster is associated with repressing pigmentation. I can't say that this is certainly the case in the montium subgroup, but it's a very close relative, and we can at least say for now that PDM3 is capable of regulating pigmentation. So getting back to these top 2 SNPs, I wanted to know what was happening between this red and blue associated SNPs, since they occurred only 480 bases apart from each other. So what I found was an interesting mapping bias, where reads from the light lines mapped very well to the reference genome within this region, whereas reads from the dark lines did not. And so in this integrative genome viewer plot, these red outlined reads show reads where one pair in a mate, or in a paired end read is mapping and the other partner is not. So I suggested maybe there's some kind of rearrangement here. So I took all the reads from the dark line and reconstructed a haplotype that I'm showing you here in this cartoon, where the red and blue lines represent the SNPs that we found, and you can see that in the intervening regions, there are two different alleles of different sizes. So the dark allele is 333 bases longer. I put PCR primers here, shown by these arrows, and what I found is that there are definitely two discrete alleles. So light flies are homozygous for the shorter allele, and dark flies are homozygous for the longer allele. So what is in this region? When I line the two sequences together with a dot plot and putting a dot every time the two sequences share 90% of a match, what you can see is that the flanking regions look very similar to each other in both alleles, but the intermediate region really doesn't share very much sequence homology. However, there is a hint that maybe there might be some repeated elements in this region, potentially formed by tandem duplications. So to look at this a little further, I used the meme suite TomTom tools to look for enrichment of particular motifs. And I found that 3 motifs seem to be repeated in the dark allele that were absent in the light allele. And that these were repeated in sets of 3-4 times, so perfectly fitting with my idea that there might be tandem duplications in this region. And I'm showing the red and blue SNP for reference here that we found in our genome association study. And most excitingly, motif number 2 contains predicted binding sites for abdominal B, the Hox gene regulator in Drosophila, that's really important in abdominal development, and double sex, the main sexual differentiation gene in Drosophila. So I couldn't have asked for better candidate binding motifs, and I'm currently exploring how these are regulating female pigmentation here. But my current hypothesis is that maybe female-limited color dimorphism in Drosophila serrata has evolved via the activator model, where the dark allele has gained abdominal B and double sex binding sites, and potentially males never had to express dark pigmentation. But as I mentioned earlier, there is a great diversity of pigmentation in this Drosophila montium subgroup. So we teamed up with John Pola and Amir Yassin at the University of Wisconsin, who had developed introgression mapping lines to study pigmentation in these 3 other species shown here in red. So as before, we backcross these flies into the light background, sequence the parental genomes as well as the hybrids introgressions, and then what we could do is ask where are the alleles most different. And what you can see in these 3 plots on the left is that there is an excess diversity or divergence between the light and dark strains in chromosome 2R, containing the gene PDM3 in this. However, if we look at this locus and sequence and compare alleles phylogeny across the region that was important for serrata, we don't see a grouping of the dark alleles. So dark serrata alleles and light serrata alleles are sort of forming 2 clades, but they're not forming one monophyletic clade with all the dark alleles. Furthermore, if you look at the SNPs that were important in serrata, shown here by these light red and blue SNPs that were important in serrata, you can see that in Kikoi and Leontia, at least in these species, that these triangles are representing sort of the SNPs that seem to be most important or associated. And so we think that there are multiple origins of PDM3 alleles in these species. So I think it's an interesting case where potentially repeated use of PDM3 has been important, but potentially different alleles have arisen to give rise to this trait. So getting back to my main question, how frequently does each model occur? We're beginning to answer this question. So I think in Drosophila serrata, it's potentially evolving via the activator model. But as we get down to the nucleotides that are important in Drosophila kikoi and Leontia and Burlei, I think we'll be able to answer more definitively whether we think it's evolving via the activator or silencer model. And one idea I have is that maybe Drosophila burlei, where males are dark, unlike these other species, potentially a different model might be important for that evolution. We can also ask how do alleles become sex-specific. So in Drosophila serrata, it seems that abdominal B and double sex sites might be important. So I'm currently testing that, but it'll be interesting to see whether these types of motifs are important in the evolution of this trait for female-specific pigmentation in these other species. So I'm really interested in trying to figure out what is actually, at the nucleotide level, responsible for making a sexually dimorphic allele. So I'd like to thank my co-authors, all the other members of the COP Lab, undergraduates, and our funding sources. Thanks. Happy to take questions. Yeah?
So you see this repeat of like double sex the continuous double sex sites. Does that occur elsewhere in the genome? Like, is that like some kind of genome-wide repeat?
I haven't seen that, but I haven't tested directly how frequently you would find this. Yeah. Yes.
Do the transcription factors have the identical binding specificity in all the species, or is that no?
So these are predictions based on Drosophila melanogaster. So I think it's really worth testing how is this region, is it actually being bound as predicted by these binding sites? And then, yeah, does this region have a function or influence the expression of PDM3?
That your interpretation is that the sex specificity is completely coming from the double sex binding?
That's my hypothesis right now, but we're testing that.